Charles William Amir Mansour here, scientist and global business development manager for Agilent Technologies. In this short video, I'd like to walk you through one of many target applications driven by our quantitative imaging and microscopy solutions. Collective cell migration is integral to numerous critical physiological processes, including morphogenesis and tissue regeneration, and has also been identified as playing a major role in metastatic disease. The wound healing or scratch assay is one of the most highly used in vitro methods to monitor and quantify collective cell migration. The current standard involves manual wound creation, which yields low reproducibility between wounds, high variability within generated data, and possible false conclusions regarding test molecules. Using an automated wound creation tool, in addition to kinetic image capture and analysis, repeatable wounds and robust and repeatable results are easily attained. In this application, we demonstrate the use of the Agilent Biotech AutoScratch platform, an automated tool to create scratch wounds in cell monolayers formed on the bottom of microplates. With the single push of a button and using a 4 or 8 pin head, consistent scratches of equivalent size and area are made in either 24 or 96 well plates. A multi-reservoir cleaning trough is also incorporated on the deck of the tool. Using the onboard programmed procedure, unattended cleaning and decontamination of each pin is accomplished before and after use. The small footprint permits insertion of the tool into any size laminar flow hood, enabling wound creation in a sterile manner. Following washing, which can be performed by Agilent Biotech's MultiFlow FX or 50TS washers, the plate can then be transferred to one of Agilent Biotech's automated imagers or the BioSpar 8 live cell imaging system to kinetically monitor cell migration using label-free high-contrast bright field imaging. Gen 5 then masks the area covered by the invading cells over time and calculates total area coverage, average wound width, and a normalized wound confluence measurement over time. From the kinetic curve, a maximum wound healing rate can also be calculated to completely ascertain wound healing rates of cell models and potential inhibition of cell migration of test molecules. In the study shown here, we used Auris Pro microplates sourced from Platypus Technologies to investigate cell migration using a GFP-expressing breast cancer cell line. The plate contains a biodegradable gel printed in the XY center of each well. When the cell suspension is added to each well, the cells settle to the bottom of the well around the gel. Shortly after this step, the gel dissolves, leaving a cell-free zone. One can then treat each well with compounds that either facilitate or inhibit cell migration. Our imaging platform is then used to calculate the percentage confluence of cells migrating into the cell-free zone using an algorithm integrated within Gen 5 software. This then allows us to assess the ability of a drug to induce or inhibit cell migration and calculate its efficacy. As with the scratch assay, Agilent Biotech automated imaging solutions support both fluorescent, labeled base, and label-free methods for quantifying cell migration within cell exclusion zone formats. Thank you for taking the time to learn more about our application workflow. Feel free to explore other application areas through this video series.